Dear students, Assalamu alaikum to all of you in 11th botany. Our today's topic is Krebs cycle, and this is a second step of anaerobic respiration. Before this, we have discussed glycolysis, and in this glycolysis, we have started from one molecule of glucose, and at last we have got two molecules of pyruvic acid, or we can say 2 pyruvate. So as per this cycle is concerned, it is also called as Krebs cycle, and it is named after the name of scientist. His name was Hans Krebs, who gave this complete cycle. And this is also called as TCA cycle, tricarboxylic acid cycle, because the first compound which is formed is citric acid, which is a 3 carboxylic acid compound, as there are 3 carboxyl groups. Now, the molecule which is going to participate here is pyruvic acid, but as such, this pyruvic acid cannot participate in this cycle or in this reaction. So, pyruvic acid changes into a 2 carbon compound which is acetyl coenzyme A, and we know that pyruvic acid is a 3 carbon compound and acetyl coenzyme A is a 2 carbon compound. Now acetyl coenzyme A participates in the reaction. During this reaction, a couple of changes will take place. As pyruvic acid, we know it is a 3 carbon compound, and one carbon compound, one CO2 is given out in the form of carbon dioxide, and secondly NAD positive gets reduced to NADH2. As it is a redox reaction, so one pyruvic acid will give us one NADH2 and another will give us next NADH2. So we will count, when we will count the energy, we have to count ATPs. When we count ATPs, we know that we have to multiply it by 2. Now in case of pyruvic acid, we know that first of all, when we take this pyruvic acid, which is a 3 carbon compound, and in this one CO2 is given out and one coenzyme is joins here, it means that it becomes acetyl coenzyme A. This acetyl coenzyme A, we know that it is a 2 carbon compound. Now acetyl coenzyme A is accepted by a 4 carbon compound which is oxaloacetic acid. Then when these combine, 2 carbons combine with 4 carbon compound, then we get the compound which is citric acid, and this citric acid is a 6 carbon compound. The enzyme which is working here is citrate synthetase. Now this citric acid, we know that it is 6 carbon compound. It changes into isocitrate, and isocitrate is also a 6 carbon compound, and here the enzyme which is working here is isomerase. This isocitrate 6 carbon compound, it changes into 5 carbon compound, and in this one carbon is given out in the form of carbon dioxide, and this 5 carbon compound is alpha ketoglutarate. Then it is again a redox reaction, so NAD positive gets reduced to NADH2. Now 5 carbon compound which is alpha glutarate, it changes into 4 carbon compound. Here one CO2 is and the compound becomes succinyl coenzyme A. Here coenzyme is added, one CO2 is given out, and one coenzyme is added. So the compound formed is succinyl coenzyme A. This is also a redox reaction. So substance getting reduced is NAD positive into NADH2. Now succinyl coenzyme A, we know that it is 4 carbon compound. It changes into simple succinic acid. That is coenzyme A will be given out. That means some energy is released in the reaction. This energy is used to convert GTP to GTP. Like we have ATP, similarly there is GTP. Ultimately gives us 1 ATP. So we know that succinic acid changes into fumarate and fumarate changes into malate. Then malate will help in regeneration of oxaloacetic acid, which is the acceptor of acetyl coenzyme A. Now we know that these both pyruvic acids, as we know it is 2 pyruvate, so both pyruvic acids get converted into acetyl coenzyme A. That means starting with one glucose, there are two Krebs cycles which take part in the reaction. So this reaction takes place in mitochondria. So again we will take an overview of this Krebs cycle. First of all, we know that we have taken 2 pyruvate, or we can say 2 pyruvic acid, which is a 3 carbon compound, and it is formed as the result of glycolysis. In cytoplasm it diffuses into mitochondria, but in mitochondria it is converted into acetyl coenzyme A. And before this, when it was pyruvic acid, it loses 1 CO2 and coenzyme joins here, and here NAD gets reduced to NADH2, then it becomes acetyl coenzyme A. Now this acetyl coenzyme A, we know that acetyl coenzyme A is 2 carbon compound and it is accepted by a 4 carbon compound, which is oxaloacetic acid. Then we get, as the result we get citric acid, or we can say citrate, which is a 6 carbon compound, and it is reaction number 1. Now the enzyme working here is citrate synthetase. Then citric acid, which is a 6 carbon compound, it changes into isocitrate. It is also a 6 carbon compound and it is reaction number 2. The enzyme which is working here is isomerase. Then isomerase, we know it is 6 carbon compound, it changes into 5 carbon compound, that is 1 carbon will be given out, as we know it is also a redox reaction. NAD positive gets reduced to NADH2. So the compound formed is alpha ketoglycerate, which is a 5 carbon compound, and it is reaction 3rd. Now 5 carbon compound alpha ketoglycerate changes into 4 carbon compound. So here one more CO2 is given out and one coenzyme is added. The compound formed is succinyl coenzyme A, which is a 4 carbon compound, and it becomes reaction 4. Again it is a redox reaction, and again in this substance gets reducing is NAD positive into NADH2. So this reaction is reaction number 4. Succinyl coenzyme A, it means that, which is a 4 carbon compound, it changes into simple succinic acid. It is reaction number 5, and this coenzyme A is given out. Then some energy is released, and this energy is released to convert GTP to GTP. Like we have ATP, similarly GTP, and this GTP gives out 1 ATP. So this reaction is reaction number 5. Then succinic acid changes into fumarate, which is reaction number 6. And this is again a redox reaction. So the substance which is getting reduced here is FAD positive to FADH2. Now we know that each NADH2 gives 3 ATPs and FADH2 will form only 2 ATPs. Now fumarate, or we can say fumaric acid, changes into malate. It means reaction number 7. Then malate changes into oxaloacetic acid, which is reaction number 8. So it is also a redox reaction. being redox reaction, here NAD gets reduced to NADH2. So malate, in this way malate helps us in regeneration of oxaloacetic acid, which is an acceptor of acetyl coenzyme A. Now we have to count the energy, and we have to see how many ADPs are here. So we have to see how many NADH2s are here. So we have to see how many FADHs are here, how many ATPs are here. Now here we know that there are 3 NADH2s. It means that one NADH2 is equal to its worth is 3 ATPs. Each NADH2's worth is 3 ATPs. So it means that when there are 3 NADH2s, it means 3 into 3, which becomes 9. Now here is one FADH2 and one FAD worth is 2 ATPs. So one ATP is here only. So it means there are 2 ATPs. It means that 9 plus 2. Then there is one ATP. So ATP is only one ATP. It means that 9 plus 2 plus 1. It means that total they become 12 ATPs. So starting with glucose, it is only one Krebs cycle. It becomes 12 ATPs. But starting with glucose, we have two pyruvic acids. So we have to multiply it with 2. So it becomes 12 into 2. It means it becomes 24. So if the question is asked about one Krebs cycle, then it is 12 ATPs. So if we are asked how many ATPs are here starting with glucose, then there are 2 Krebs cycles. That means 12 is multiplied by 2, it becomes 24 ATPs. Now I will give here a shortcut sentence by which you can remember these substances. So some shortcut sentence by which you can remember is: see, I know some simple formula making options. C starts from C. It means that C refers to citric acid. Then there is I refers to isocitrate. Then there is no starts from K. It means that alpha ketoglycerate. Then there is some refers to succinyl coenzyme A. Then there is simple refers to succinic acid. Then there is a formula, F refers to fumarate. Then there is making, M refers to malate. Then there is options, O refers to oxaloacetic acid. So it was all about Krebs cycle. Thank you so much.